Anthropometry or Berthollet system. It is the science dealing with the measurement of the human body to determine the differences in individuals. It was introduced by Alphonse Bertillon in 1900s. The composition of Bertillon system. It has three compositions. The first is anthropometrical signalment. This is the measurement of the body, head, and limbs. For example, second one is descriptive signalment, or a clear specific system of description of an individual as well as the photography of the male factor. And the last one is peculiar mark signalment. It is by indelible marks, for example, scars, tattooing, birthmarks, moles, and etc. Did you know that forensic anthropology can be described as the analysis of human remains? It is used for medical legal purpose of establishing identity. So, forensic anthropology is a special subfield of physical anthropology. Anthropology means the study of human remains. That involves applying skeletal analysis and techniques in archaeology to solving crime and criminal cases. There are four basic criteria for the basis of any primary identification. First one, the skeletal or developmental age. The second one is biological or sex. Number 3, living stature. And last one is ancestry or racial affiliation. The bones we're interested in personal identification are number 1, skull. Number 2, humerus, femur, and tibia or the long bones, and the last is pelvis. Word portrait parle in 1882 or the speaking likeness is a verbal and pictorial description of a person being identified. Such information may be given by witness, relatives, or a person who is acquainted with physical features of the person to be identified. For example, the face, skin color, hair type, eyes, eye colors, and the color of the eyebrows, and etc. Odontology is the scientific study of the structure and disease of teeth. Forensic odontology, it is the proper collection, interpretation, evaluation, as well as presentation in court of dental evidence during the conduct of criminal and civil proceedings. And it is the application of dentistry in the legal proceeding deriving from any evidence that pertains to teeth. White teeth. White teeth. Every human body ages in a similar manner. The teeth also follows a semi-standardized pattern. These quantitative measurements help establish relative age of a person. Each human has an individual set of teeth which can be traced back to establish the dental records to find missing individual. Teeth is made up of enamel. Enamel is the hardest tissue of the body, so it can withstand trauma better than other tissues in the body. Teeth are sources of DNA. Dental pulp or a crushed tooth can provide nuclear or mitochondrial DNA that helps to identify a person. It has approximately 32 teeth in adult mouth. There are 4 types of teeth. The number 1 is molars, number 2 premolars, number 3 canine, and number 4 incisors. The teeth differs in size, shape, and root type. The teeth through the years.
Primary teeth sprout from milk buds and are temporary. Once they fall out, permanent teeth as seen on the other side appear. Adulthood, 21 to 25 years old. Permanent adult teeth come in when primary teeth fall out. They are permanent because they establish roots inside the gums. Third molar comes in around the mid-teenage years.
Bite marks.
Bite marks are impressions from teeth found on skin or items left at a scene. This is usually outline teeth placement.
Analyzing bite marks. Bite marks are photographed with a scale. Bite marks on skin are taken over repeated intervals. Casts of impression are taken. Impression trace into transparencies. Casts of suspect teeth are taken. Comparison between suspect casts and bite marks.
Various bite marks.
Forensic serology. Serology is the study of bodily fluids such as blood, urine, saliva, semen, and sweat. Forensic serology to the scientific analysis of the blood and other bodily fluids as evidence during the conduct of criminal investigation. The serology section of forensic laboratory may deal with any or all of the following: blood typing, characterization of unknown stains, blood spatter analysis for crime reconstruction, paternity testing, semen identification in sexual assault cases, DNA typing for identification of victims and the suspect. The composition of blood. Blood is a mixture of many components, cells, enzymes, proteins, inorganic substance, or salt, water. 55% of blood content is plasma, mostly water and substance dissolved in it. Most of solid materials are red blood cells or erythrocytes, white blood cells or leukocytes. Forensic characterization of bloodstains. 3 questions that must be answered by forensic investigator. 1. Is it blood? 2. If yes, is it human blood? 3. If yes, can it be associated with individuals? For the question of is it blood, presumptive test for blood. Presumptive tests rely on hemoglobin's ability to catalyze the oxidation of certain reagents, usually resulting in a color change. Oxidizing agent is usually hydrogen peroxide or H2O2. For the visible blood stain, Castle-Meyer solution of phenolphthalein is applied to suspect blood stain, followed by hydrogen peroxide. A bright pink color indicates the presence of blood. For example,
the Kastle-Meyer test, or phenolphthalein test, is a presumptive test for the presence of blood. This is a 3-step test. First, take a sample of the blood stain to be tested by using a cotton swab or the corner of a piece of filter paper. Only a very small sample of the stain is needed for testing. To swab, add a drop or 2 of ethanol. This is followed by a drop or 2 of Kastle-Meyer reagent. Finally, add a drop or 2 of 3% hydrogen peroxide. A positive result will be indicated by the immediate presence of a pink color. There will be no...
The next one, the leukomalachite green test. Same procedure as Castle-Meyer. Procedures blue-green color in presence of blood. For example again. For the invisible blood stains, luminol test used for the large areas where blood is suspected but not visible. If the blood is present, it will glow, must be viewed in the dark and photographed quickly. And for the example is now, if you know that is it blood, the next step is confirmatory test. Once a presumptive test indicates that stain may be blood, the serologist must confirm that is it human blood. The test must be precipitin test. Precipitin test identifies the presence of proteins that are found only in human blood. Blood typing antigens. Antigens allow a living system to recognize foreign biological substances such as infectious agents and allergens. Antigens also impact specific characteristics to the red blood cells. These characteristics can be partly used to identify individuals.
Acid phosphatase is an enzyme secreted by the prostate gland that is present in large amounts in seminal fluid. It is not unique to the prostate and can be found in other biological fluids, including vaginal secretions. It is therefore considered a presumptive chemical test for the presence of semen, and semen must be confirmed by other means.
Take a sheet of filter paper and moisten. Place over the suspected stain and mark the position and hold in place for about 1 minute. Remove the filter paper and take the acid phosphatase reagent and spray on the filter paper. A purple colour change within 1 to 2 minutes indicates a positive reaction for semen.
DNA fingerprinting. DNA stands for deoxyribonucleic acid. DNA fingerprinting reported by English geneticist Alec Jeffreys, who noted that certain DNA sequences were repeated over and over again next to each other in certain DNA regions. DNA is a genetic material found within the cell nuclei of all living things. With the exception of identical twins, the complete DNA of each individual is unique. DNA looks like an incredibly long twisted ladder. This shape is called a double helix. The rungs connected to the carbon sugars, also known as bases. These are the 4 bases: A for adenine, T for thymine, G for guanine, and C for cytosine. They may consist of millions of base pairs.
Step 1. Isolation of DNA. DNA must be recovered from cells or tissue. Only a small amount of blood, hair, or skin is needed to isolate DNA. Step 2. Digestion of DNA by restriction and the nucleases. The DNA is cut into fragments using restriction enzymes. Each restriction enzyme cuts DNA at a specific base sequence. Step 3. Electrophoretic separation of different fragments. Fragments are separated on the basis of size using a process of gel electrophoresis. The shorter DNA fragments move faster than the longest fragments. Step 4. Transfer DNA on nylon or nitrocellulose membrane. The DNA fragments are transferred to nylon sheet by placing the sheet on the gel and soaking them overnight by the process of southern blot. Step 5. Probing or probe labeling. Adding radioactive or colored probes to the nylon sheet, which is complementary to target sequence. Step 6. Hybridization. Labeled probe DNA should be hybridized with the complementary sequence located on nylon membrane for the detection of position of latter membrane is washed to remove non-specific binding and clearing of the background. Step 7. Autoradiography. Hybridized membrane with X-ray film. X-ray film alter is a different shows multiple number of bands that looks like barcode and known as DNA fingerprints. Step 8. Interpretation of band patterns.